Hello students, how are you people? I hope you people are all fine and healthy. Okay, with the next topic of the day, that is the resting membrane potential. We discussed, I gave you the introduction of the active membrane potential. This is the board of the last lecture. Go to my previous lecture for detail of this lecture. We are going to start the resting membrane potential. Today we will be discussing the features of the resting membrane potential, the characteristics of the resting membrane potential. Actually, what is a resting membrane potential? A resting membrane potential is the average distribution. It is the average distribution of charges across. It is the average distribution of charges across the axonal membrane. It is the distribution of charges across the axonal membrane, right? Distribution of charges across the axonal membrane that keeps them separate during our non-conducting conditions, when the neuron is not in a conducting state. It is not conducting any message. It is not conducting any impulse. To maintain the resting membrane potential, that is to keep a neuron in a resting state where it is not conducting any potential, it is not conducting any message, what do we have to do? We have to study the conditions. Now, when a membrane is in a resting membrane state, when it is in a non-conducting state, we say that it is in a polarized state. Remember this. The membrane in an RMP is in a polarized state. It is said to be polarized, and the neuron in this is supposed to be, it has two different charges across each side. Now, if this is the neuron, let me draw a diagram for you people. If this is the neuron membrane, if this is the neuron, yes, this and this. If this is a neuron, we have distribution of charges inside and outside. This distribution of charges inside and out, this is the distribution of charges that we have in a neuron when it is in a polar state. A complete distribution of charges across the membrane. A complete distribution of charges across the membrane. Look at this. Positive and out. So we see that we have a complete negative charge inside and a complete positive charge. Now, these two different charges are totally separated from each other across the membrane. This is said to be in a polarized state. In neuron in this condition, it is said to be in a polarized state. And the magnitude of the voltage that is present in this state is said to be around 70 millivolts. 70 millivolts, and we represent it by a negative sign. We call it minus 70 millivolts. But this is not the magnitude. It is not the magnitude. It is only the specification of the presence of the negative charge. Specification of the presence of the negative charge. The negative charge is present inside. When the negative charge is present inside, then it means that we will show a negative charge with the voltage that we are measuring. We will show a negative charge with the voltage that we are measuring. If the negative charge is present outside, then the condition is going to be reversed. Then we will write it as a positive voltage. Here, we are writing it as a negative voltage because the negative charge is present inside. Okay, it is present inside. Right. Now, here, what do we have? The next condition that I'm going to tell you, this is the amount of voltage that is present across. Okay. Now, the next condition that I am going to discuss, this is a situation that the membrane is in a resting membrane potential. Then the neuron is in the resting membrane potential. Now, the conditions that will maintain this neuron in this state, in a non-distribution state, this is very, very important. First of all, we are going to see the concentration of the sodium, the potassium ions inside the neuron, the sodium and the potassium ions. There is a specific concentration of the sodium and the potassium ions. Now, this is the internal area and this is the external area of the neuron. This is an environment that is present inside, and these two, sodium and potassium, they are present in a specific balance. They are present in a specific balance, and in a specific balance, what happens? In a specific balance, they are present in a specific concentration. For example, potassium is 30 times greater inside. Potassium is 30 times greater inside as compared to outside. 30 is to 1 is going to be the ratio of potassium in and out. This is going to be the ratio of potassium in a neuron in a resting state. And sodium is 10 times greater outside the membrane. Sodium is going to be 10 times, it is 10 times greater outside the membrane as compared to inside. Sodium, that means that sodium is going to be 10 times greater as compared to the inside. That is, it is going to be 10 is to 1. Now here, this is the place where we are going to have 30 potassium. We are going to have 30 potassium over here, 30 potassium inside, and 1 sodium. Over here outside, we are going to have outside here, we are going to have 1 potassium and 10 sodium. So this is the ratio that is existing across the membrane in a resting membrane situation, in a resting membrane state. This ratio has to be maintained. If this ratio is not maintained, then the polarity is going to be disturbed, and then the neuron will not be in a conducting state. These ions, what do? These ions, they are continuously moving against a gradient. Moving against a gradient means that they are going to follow the active transport. They are going to follow the active transport. Moving against the gradient means that more potassium is going to come inside and more sodium is going to move outside across the sodium potassium pump. In the previous class, I told you about the sodium potassium pump. It is going to use energy. There is going to be expenditure of energy. Expenditure of energy in what form? Expenditure of energy in the form of ATP. It is going to be expenditure of energy in the form of ATP. 2 potassium are going to be actively transported inside. How many potassium? 2 potassium. They are going to be transported inwards. Potassium is in a high concentration, and then energy is going to push in more. And 3 sodium as compared to 2 potassium. 3 sodium are going to be actively transported out. This is what is happening in the active membrane state. 3 sodium are going to be transported out. They're going to be transported out of the membrane. And because of this, what happens? We see the development of a more negative state inside, which gives us the accumulation of negative charge inside. Now, when we come to the negative charge, the negative organic ions, there are many types of negative organic ions in the neuron, in the neuron cytoplasm. And these negative ions, they are actually, these negative ions, they can be amino acids, different kinds of proteins, amino acids, different kinds of proteins. Then we have RNAs. These are all present inside inside the neuron as compared to the outside. So it is rendering the inside more negative as compared to the outside. We are discussing the resting membrane potential when the neuron is in a not conducting state, when the neuron is not conducting any message, when the neuron is resting. There are more negative ions inside as compared to outside. There are more positive ions moving out. Except movement through the pump, we have channels also. And there is going to be a leakage. There is going to be a leakage of potassium. from these channels. There is going to be a leakage of potassium from these channels, and because of that, more positive is moving out. ठीक है, the leakage of potassium across the channel का मतलब ये है कि वो it is going to be according to the gradient. Now overall, more charges are moving outside, and the more charges will be present on the outside as compared to the inside. Therefore, the inside and outside is going to be differentially polarized. It is going to have a different charge, and this is a situation in which a neuron is said to be in a polarized state. Right? Now, to disturb this distribution, what is required? A stimulus is required, and that stimulus must be of a specific power. It should be of a specific strength. Should be of a specific strength, and that specific strength is known as the threshold. The minimum power of the stimulus that is required to disturb the distribution across the membrane is known as the threshold. If the threshold is strong enough, then that is said to be appropriate. That is said to be enough to disturb the distribution. That is called the adequate stimulus. If the threshold is strong enough to disturb this distribution, then it is called the adequate stimulus. It is called the adequate stimulus. It is enough to disturb the distribution and bring the resting membrane potential into the active membrane potential. If the threshold is not strong enough, then it is said to be subadequate or subthreshold. It is said to be subthreshold. Threshold is not complete to disturb this distribution. And what is the distribution? 30:1 potassium, 1:10 sodium. Active transport through the pumps, passive transport through the channels of potassium, more concentration of negative ions inside, minus 70 millivolt distribution inside. Let me go to the presentation now. Let us have a reading of the presentation, and then we will close the class for the next lecture. Nerve impulse, we have already discussed this. It is a wave of electrochemical changes along the length of the neuron. Electrical potential is the measure of capacity to do the electrical work. Electrical potential exists along the membrane, and it is a membrane potential. The resting membrane potential is the net difference in charge between the inner and the outer surface of a non-conducting neuron. The major factors that are maintaining the resting membrane potential, I have already told you, are the sodium ions, the negative organic ions, amino acids, proteins, RNAs, leakage of potassium from the neuron through the channels, no conduction of nerve impulse. The membrane potential is going to be minus 70 millivolts or minus 0.07 volts. It exists across the membrane. This is the potential which is going to separate the charges across the membrane. Again, nerve impulse is an electrochemical change along the length of a neuron and involves the movement of ions across the cell membrane. I repeat these things over and over again so that all the students that are attending the lecture, that are watching the lecture, they should get an idea of what I am trying to teach. The neuron membrane is positively charged on the outer side and negatively charged on the inner side, and it has a specific electrical potential. Electrical potential, what is it? Is the measure of capacity to do electrical work. It represents a type of stored energy, a type of potential energy, which is used for separation of charges across the barrier. Electrical potential across a membrane is known as the membrane potential. The net difference in charge between the outer and the inner surface of a non-conducting membrane is a neuron is known as a resting membrane potential. It is known as the resting membrane potential. This is the extracellular fluid. This is the intracellular fluid. And look here, we have more negative accumulated close to the membrane and more positive accumulated outside. So this is in the resting membrane potential. Now look here. 2 potassium are moving in through the channel. 3 sodium are moving out, through the pump. 3 sodium are moving out through the pump. This is the exterior. This is the interior. sodium is diffusing passively inside, potassium is diffusing passively outside, but more potassium is diffusing, so the arrow is larger in size and less sodium is coming in. And because of this, more positive charges are moving out. And because more positive charges are moving out, this is going to have a negative accumulation of charges inside as compared to outside. The major factors in the resting membrane potential: sodium is 10-fold outside and potassium is 30-fold. Sorry, this is somebody, I wrote 20 here by mistake. This is 30, this is not 20, okay? Potassium is 20 times more inside. As sodium potassium pumps, 3 sodium move outward for 2 potassium which come inward. The potassium also diffuses through the membrane ion channels, leaving the inside negatively charged due to negative ions like amino acids, RNAs, proteins, phosphates, etc., etc. Now, this is the situation. 3 sodium out. This is sodium. This is potassium. This is a sodium potassium pump. This is a potassium channel. This is a sodium channel. Now you can see that passive movement across the channels. Passive movement across the channels. Active movement across the pump, where ATP is going to be utilized. Now, this is the concentration. 30 potassium inside is to 1 outside. 10 sodium outside is to 1 inside. 3 sodium shifted out, 2 potassium coming in. Leakage of potassium towards the exterior. Less incoming of sodium towards from the exterior to the interior. More potassium is lost. 1, 2, 3, 4 channels. 1 channel of sodium. So we are seeing the loss of more positive charges. Accumulation of negative ions inside. Negative ions are not moving out as compared to outside. That is rendering the inner more negative. The negative ions and inorganic ions are much more inside than outside. Leakage of potassium takes place continuously, making the inside more negative. No conduction of nerve impulse. The resting membrane potential count is 0.07 volts, minus 0.07, just to show the presence of negative charge. If the negative charge is inside, it is negative, more negative inside, then we write the negative with the RMP voltage. If negative is outside, then we will write positive. It is not the magnitude, it is just telling us the position of the negative charges. Now, in the next class, we will be discussing the nerve impulse, how the nerve impulse is going to be initiated. It is going to be initiated by an appropriate threshold. In the next class, we will start from the threshold and we will move on to the next lecture. This was all for today. I hope you understood the lecture. If you have any question, don't hesitate to ask me. See you people next time. Take care of yourself.